Welcome to our final video on topic 5 of evolution and biodiversity. In this video, 5.4, we're looking at cladistics. Now, the whole essential idea of this topic of cladistics is that the ancestry of a group of species can be deduced by comparing their base or amino acid sequences. So we're really looking at that whole molecular concept of the makeup of the DNA of the organism. So our understandings. First of all, we need to have a look at what a clade is. And a clade is an infographic, as you can see here, where we look at ancestral pathways. So a group of organisms that have evolved from a common ancestor. Okay, so that is our clade. They include all species alive today, but also includes common ancestral species and any species that has evolved from it and then may have become extinct. Now, if you look at the top two diagrams, which are showing clades, okay, you'll see that each group there has a common ancestor. Now, the two that are representing examples that are not clades, you'll see that group there has a different ancestor to this group over here that's ancestor is there. Okay, and the same here, this is showing two different ancestral pathways, even though the initial ancestor was the same, we've branched out into other ancestral pathways. So our clade follows the exact ancestors. All right. Now, clades are groups of taxa together according to the characteristics that they have evolved most recently. Now, cladistics is therefore an example of natural classification. We looked at natural classification in our previous video. Now, when we're looking at cladistics, we look at these things called cladograms. Okay, and these are tree diagrams showing the similarities and differences between our species. So these tree diagrams, noting that any points where there's a branch, we call them nodes. Okay, and nodes denote speciation. So we talked about this in a way earlier video, but we didn't call these cladograms at that point. Now, cladograms are based on DNA sequencing as well, okay? So cladograms are tree diagrams, as we've just said, that show the most probable sequence of divergence in clades. Cladograms are based on the similarities and differences between species in a clade, as we just spoke about. Okay, and we're just showing two different kind of cladograms here as such. Both of them having those common ancestral points. Okay, so our nodes. Yeah, just two different ways of viewing it. So what biochemical evidence is there to support this theory of the clade and clades? Well, the discovery of DNA and protein structures has really brought about the validity to this theory, okay, and the confirmation of the idea that there are common ancestors. For example, the fact that every known living organism on Earth uses DNA as its main source of genetic information is compelling evidence that all life on Earth has that common ancestor. We look at this diagram here, we're showing that common ancestor, and we're looking at these base sequences here that we spoke about back in unit topic 2. Okay, and you'll see that after 25 million years, there was a change in the base sequence of both of from the common ancestor. Okay, and that's where our divergence started to occur. So when we're looking at these base sequences and the change into them is where we start looking at what we call the molecular clock. And this is where we're estimating the time since two species diverged from a common ancestor. Okay, and it's based on their differences in their biological makeup. So when DNA mutates, a particular protein then changes over time at approximately the same rate in each evolutionary line. Therefore, the molecular clock can be used to identify evolutionary relationships and to estimate when various modern species last shared a common ancestor. The clock ticks, as such, at different rates for different proteins. The rate of change of a particular protein can differ between groups of organisms. And here we've got a diagram. So this is all about you guys learning to read different types of graphical information. So at the top, you've got a range of species, okay, on your y-axis, and then along the x-axis you've got average percentage difference in the protein chains. Okay, so between the chimp and the human there's less than 5% difference. Between the chimp and human and the elephant we've got 18% difference. Okay, now we move then B here, also an x-axis scale, is an uncorrected time scale in millions of years. So basically what you've got to imagine is if you move that scale up, okay, you can use that along the base of the graph. And then we've got, based on fossil evidence, we've got the correct time scale, okay, or the corrected time scale. So you can also use that along the same scale at the bottom there. Okay, so if we're looking at when the elephant and human, or the human and chimp and the elephant diverged, on the uncorrected scale, we'd be looking at about 125 and on million years ago, and on the corrected scale, we're looking at 65 million years ago. So quite a difference there, but that is due to the fossil record that's been developed. Alrighty, so we're thinking of this whole idea of the DNA, okay, and looking at the ancestral DNA. All right, so we've already touched on this whole concept of homologous and analogous features. Okay, your homologous features being the same structure but different roles, and the analogous feature being the same role but different features. So homology, this is where organisms descended from a common ancestor and show similarities in structure regardless of their way of life. Similarity similarity in basic structure exists even though these structures now serve a different function. We call these homologous structures, remember. And then we've got our analogy, where structures that carry out similar function but are not necessarily similar in basic structure. So they're derived from different ancestral structures. Organisms cannot have arisen from a common ancestor if it's an analogous structure that we're looking at. Okay, so we've got some homologous features, analogous features. So homologous, the form is similar, analogous, the form is different. With function, homologous, the feature is different, but in analogous, the function is similar. We've got our examples there, and used as evidence of common ancestry, yes for homologous, no for analogous. Alrighty, cladograms and reclassification. So this whole concept of reclassifying due to new scientific developments as such. Okay, so problems in distinguishing between homologous and analogous structures have sometimes led to mistakes of classification being made in the past. For this reason, morphology, so the form and structure of an organism, is rarely used nowadays for identifying members of a clade. Molecular evidence is now much more commonly used. Evidence from cladistics has shown that classification of some groups based on structure did not correspond with the evolutionary origins of a group or species, and as a result, some groups have been reclassified. Reclassification of groups of organisms is time-consuming and potentially disruptive to biologists. But it needs to be done because as we're finding these new, having new developments in scientific knowledge, this kind of thing is going to happen more and more. All right, so looking at our applications and skills now. You need to be able to recognize some cladograms that include humans in them. So we've got a couple of examples there. Knowing that humans are most closely related to the chimpanzee. Okay, gorillas right next to us there as well. And our least close relation as such would be the lemurs. Got another example of a cladogram here with humans on the right hand side, lemurs right down the other end. Okay, another application and skill is based around you being able to recognise the falsification of theories, with one theory being suppressed by another. And an example of this is seen in the plant families that have been reclassified as a result of evidence from the cladistics. Now, classification of our angiospermophytes, so our flowering plants, remember, into families based alone on their morphology was conducted way back in 1789 by French botanist Antoine Laurent de Jazzy. Taxonomists have recently investigated the evolutionary origins of the figwort family and using cladistics and compared to base sequences of different chloroplast genes. As a result of this comparison, it was found that the figwort family were not a true clade and that 5 clades had incorrectly been combined into one family. A major reclassification of the figworts was then required. Okay, so having a bit of a look at this use of clades in practice. Okay, so let's have a look at the cladogram in the picture here. And first of all, what animal is most closely related to the squirrel? So find the squirrel and note down what animal is closely related. We'll come to the answers in a minute. Number 2, what animals are more closely related to humans than old world monkeys? So find a human, find the old world monkeys, and who are closer related to the humans than the old world monkey? The tarsiers share a common ancestor with which animal? So find the tarsier, and then which animals share that common ancestor? We'll look at the answers in a minute. Write them down. All right, elephant shrews, elephants, manatee, and the dugong all share a common ancestor. Which animal diverged from the ancestor first? Alrighty, let's have a look at our answers. First up, the animal most closely related to the squirrel is the guinea pig. Number 2, what animals are more closely related to humans than old world monkeys? We've got the gibbons, the orangutans, the gorillas, the baboons, and the bonobos and the common chimpanzees. They're all closer to humans than the old world monkey. Okay, the tarsier share a common ancestor with which animals? They've got common ancestors. We've got the new world monkey, the old world monkey, the gibbons, the orangutans, the gorilla, the humans, the bonobos, and the common chimpanzee. So we're looking at the tarsier, and then it'll be all of these listed here. Elephant shrews, elephants, manatee, and the dugong all share a common ancestor. Which animal diverged first? The elephant shrew. So here we are. We've got all of these 3 here, and the elephant shrew is the first one to diverge. Okay, we've got another cladogram here of 4 genera of plants. So first of all, you've got to name the 2 sister taxa. That'd be the Digitalis and the Veronica. Name the outgroup in the cladogram. So that will be Antiherum up there because they are the outgroup. The rest have common ancestry. Using a clearly marked label, indicate a node. Okay, so there are nodes. And which genus possesses characteristics that evolved more recently? The Digitalis or the Plantago? And it will be the Digitalis because they have evolved from a common ancestor more recently. Okay, study the phylogenetic tree below, so your cladogram below showing some primates and their chromosome numbers. Note that when there is a great variety between one species and another, within a taxon, a range of chromosome numbers are given. Okay, so A, identify the numbered arrow that indicates a common ancestor for all the primates shown. So the common ancestor for them all would be number 1. Okay, arrow number 1. Monkeys have tails, whereas apes do not. Arrow number 3 shows the point where primates lost their tails. List the apes shown in the diagram. So number 3, this is where they lost their tails. So all primates with tails would be lemurs, monkey squirrels and baboons. Everything from number 3 onwards have lost their tails. C. Identify the numbered arrow that indicates where bipedalism completely replaced walking on 4 legs. Alrighty, completely replaced, I would say is number 4, because I think chimpanzees still sometimes walk upon 4 legs. The great apes are the 4 primates shown that demonstrate the most recently developed derived traits. Identify which taxon in the diagram represents the lesser apes. I think that is referring to arrow number 3. I think that's what that one is referring to. E. All the great apes shown except one have the same number of chromosomes. Which species has a different number of chromosomes? Okay, so yes, it was number 3, and the different number of chromosomes, sorry, it was 3, yep, on that last one, the different number of chromosomes is the humans with 46. The rest of the great apes have 48. Some evidence support the idea that in humans, 2 of our chromosomes fused together at some point in our evolution. What evidence is there in the cladogram to support this? I would say the fact that we've got 46 rather than 48. And that would have been at this point here, where the chimpanzees still have 48 and we've got 46. We've now covered all our sections of 5.4. Thank you for watching. Hope you've enjoyed this series on evolution and biodiversity, and remember to subscribe to my YouTube channel. Thank you for watching.